Welcome back, welcome back, welcome back to science. All right, so today's topic that we're looking at here, 3.5, control of cell division, is what stops a cell from growing and just growing and growing and growing into more and more and more cells. There are things that regulate when they replicate and when they stop replicating, and that's this. And also, we're going to look at well, what happens if they lose the ability to hear the signal of stop, just wait, we're good. We're also going to look at that. All right. Certain cells reproduce all the time. Your skin cells, your intestinal cells being bathed with those digestive enzymes, cells in your mouth, just digestive tract from mouth all the way through anus. Oftentimes those cells reproduce in high number just due to what they're being exposed to and the job that they're doing. Also, blood cell formation is something that we continually do. But there are other cell types, like let's say a neuron, where it's going to replicate some, but then it's pretty much going to stop after that. At the very end of our DNA is a small section that's going to act as the primer to replicate the rest of the DNA. When that little primer goes through the replication process, I can use our DNA replication can't start right here and just go easily. Instead, it has to have a little section that pops off and it's going to tell it where to start. And so this red represents a primer of where to start. And so it's going to start DNA replication, and whenever it's all the way done with DNA replication, this part falls off, and then there's an unpaired group. That unpaired group won't last like that. So this unpaired piece will be broken off. That little piece right there that's broken off at the very tip of your chromosome is called a telomere. Every time that your cell replicates, a telomere will be lost off of that. The older the cell, the more pieces that have been removed. And in essence, this causes us to have mortality. It limits as long the life we can live. We can't live forever. And I know we've said that and we've heard that and stuff, but you're like, but science, you just wait. Well, science has to fix this problem, and that's the telomere. It's that piece at the very tip of the chromosome on each end that with every replication, the DNA gets a little bit shorter. And so it creates a finite life to the DNA. Essentially, it can only do that until it gets to the point that it's interfering with a gene. When that gene can't function, it may be the thing that causes disease or may even terminate life. So those tips are called telomeres. So in essence, by it shortening each time, you can look at DNA and say this is shorter, and you can determine essentially the number of replications a cell has went through as a result. It becomes in essence a mitotic clock. Because if you look at just how many pieces are missing, you can determine how many replications it's been through. With the cell cycle, different signals can signal the cell when to go through the cycle and make more, like growth hormone can trigger when to make more, or platelet-derived growth factor, PDGF, can say, hey, there's an injury, we need to make more. Or an internal regulator, such as a protein called cyclin, can trigger when a cell is told to replicate. The reasons that cells go through replication, of course, is to growth, repair, but if you think of why a cell isn't infinite big, and that is due to surface area to volume ratio. The larger the cell, the more volume per ratio within. Therefore, cells have to be small as a result. Going further, different hormones and growth factors like cyclins and platelet-derived growth factors and growth hormone, all of these are going to be involved in controls of cell division. Let's say that you have an injury on your knee, a scrape. You fell on the ice and you scraped your knee and it bled, but there's separation of tissue if you looked at it on a microscopic level. The neighboring cells are going to be triggered by platelet-derived growth factor, like, hey, you need to make more cells here. And cells are going to start to rebuild and close that gap by one cell at a time on either side. Eventually, the cells are going to touch each other. When they touch each other, the other side, it sends a stop signal. It goes through something called contact inhibition. Healthy cells are going to submit to contact inhibition and stop cell growth. When a cell doesn't respond to the signals to tell it to stop, then cells will keep growing. If there's a mutation in the DNA at a checkpoint, what's the role of a checkpoint? A checkpoint checks the DNA and makes sure that everything has replicated accurately. And when it has, green light, you can go to the next phase. Not, it's going to send it to a process called apoptosis, where the cell will then die. Checkpoints make sure everything is right. When a checkpoint is mutated, it can't check the quality control of the cell. And so the cell it makes may be some kind of damaged, altered mutant cell. Therefore, the cells that come from that are going to be mutated. The cells that come from those cells will also have those same mutations. And now those cells, essentially you're going to think of the master copy. The thing that you're copying is messed up. And so everything that comes from that will also contain those errors and problems. So cancer. Cancer is a disease of the cell cycle, and it's literally uncontrolled cell growth. When we have cells that are growing on other cells and growing into this mass, we call that a tumor. There's two types of tumors. There is benign and malignant. Benign is localized. Oftentimes they enlarge. There is a mass. There is a tumor, but it has not spread. Malignant tumors, those are the bad kind. They are invasive. They do spread. They are quote unquote cancerous tumors. The way that the cancer is fatal to a person is that it being malignant, it's going to grow into the neighboring tissue, damaging that tissue, and oftentimes changing the DNA of the neighboring tissue to also be mutant. That mutated DNA, those mutated cells don't do their job of what they do. These cells reproduce in high number, very fast. Some of them even do this exponentially. Whenever an organ has been affected to the extent, like a vital organ, that it can no longer do its job, that's where cancer is fatal. It stops a vital organ from being able to do its vital role. You may have also heard of genetic cancer, where people inherit certain genetic conditions. Within your DNA, you have genes called oncogenes. Oncogenes are genes that code for cancer. All of you have these. But you also have tumor suppressor genes. So you have genes that code for cancer, And then the tumor suppressor genes, they're pretty cool. Tumor suppressor genes, their job is to keep the cancer expressing gene from being expressed. Therefore, you don't have any cancer. But sometimes there's a problem. Like a tumor suppressor gene, like an example, something called P53. When P53 is mutated, it can't stop oncogenes from being expressed. Therefore, people with mutated tumor suppressor genes oftentimes have cancer. The genes that caused the cancer were already there. But tumor suppressor gene, it becoming mutated, created the problem. Here's an image of some normal hair-like cilia that's here. You can see, we know that cilia is going to be involved for the movement of material like mucus. And here is cancerous cells. Their cilia is not there. This cancer is malignant, and it has affected that tissue. Another characteristic of cancer is that cancer cells are really unique. And I know, like, probably my brightest students are thinking like, okay, so if these cells reproduce in high number, then every replication, their telomeres should be getting shorter. Therefore, they should replicate until they die. So cancer would eliminate itself. But that doesn't occur. Here's why. Cancer cells have the ability to make an enzyme called telomerase. Telomerase fixes the end of that chromosome to keep that cancer chromosome intact, essentially making cancer cells immortal. There's a line of cells called HeLa cells, which originated from a lady in the 1950s called Henrietta Lacks. There's a great documentary on this movie made by Oprah Winfrey is the starred character in this. But it is so good, depicting scientific ethic issues and the effect on that family. But in essence, cancer cells can reproduce uncontrollably, and they can just keep making more and more. And they can go through a process called angiogenesis. Angiogenesis is where the cancer mass can communicate with a blood vessel to create a branch and flow to it and feed direct blood to the tumor. And so now the nutrients available for those cancer cells are even greater. And so it's a mess whenever this cancerous mass. And now cancers themselves, they can become mobile and move, and they oftentimes move through blood vessels, or more commonly, our lymphatic system, which is where we typically fight off our cancer cells in our immune system. Our immune system is fighting in the lymphatic system, in our lymph. Oftentimes, those pathways become quite common, and it's why somebody with breast cancer oftentimes will develop ovarian cancer, even though those structures aren't beside each other, because you would think like proximity, it would have to be just its neighbor. But the vessels that are flowing throughout, that's how that's moving through the lymphatic system. All right, guys, we'll be back in just a moment, focusing on our next topic. So long.